Hi, my name is Nick, and this is Lab Coat of Paint. And today we'll dive into how scientists created these woolly mice that you've probably seen circulating around the internet in the past few weeks. We'll break down how they did it, the challenges they faced, and what this means for bringing extinct traits back into living species. And as always, sources are linked in the description below. So a little bit of background, the team at Colossal Research are placing a big bet in the fact that they can bring back the woolly mammoth, but not in the way that you think, not by resurrecting like you see in Jurassic Park, but rather what they deem as functional de-extinction by reintroducing the traits of the woolly mammoth in an Asian elephant. This approach aims to equip elephants with the necessary adaptations to fill a specific ecological role, as well as survive in certain climates, such as a cold climate. But this, you see, is a mammoth task. So as a proof of concept, they use mice for their short gestation period as well as established protocols. Although the mammoth and the mouse have 200 million years of evolutionary history between them, scientists have identified 10 specific mammoth-like features that may have allowed it to adapt to its cold, frigid environment at the time that are also present in mice. These genes primarily encode for hair length, hair texture, pigmentation, as well as fat metabolism. FAM83G, FZD6, TGM3, ASTN2, and KRT25 all regulate hair pattern, follicle orientation, density, and texture. FGF5, or fibroblast growth factor 5, controls the hair length by extending the growth phase. Melanocortin 1 receptor, or MC1R, regulates coat color by influencing melanin production. TGFA, KRT27 are all mammoth-specific mutations that affect hair characteristics. And finally, FABP2, or fatty acid binding protein 2, is involved in fatty acid transport, which could theoretically enhance fat storage for cold adaptation. Introducing 10 genetic edits simultaneously is a significant challenge. As such, researchers employed 3 different methodologies, all employing the CRISPR-Cas9 gene editing system in order to test their efficiency as well as assess the different results in phenotype. The first technique used electroporation in single-cell zygotes. So this technique uses electric pulses to deliver all of the CRISPR-Cas9 components as well as the single guide RNA, which allows it to hone into a specific site for genetic editing into the single-cell zygote. While this method boasts a high success rate, it does have the challenge of mosaicism, where not all of the cells are edited. Some may be edited, some may not. Overall, mice edited with this method showed a longer and golden fur coat with some variation on the coat texture depending on which genes were targeted. Interestingly, but expectedly, the albino mice that underwent the same editing procedure had similar hair modifications but sustained their white coat, even with disruptions to the MSR1 gene, which is responsible for coat color. The second method uses a cytosine to thymidine editor. It still uses the CRISPR-Cas9, but instead of making double-stranded DNA breaks, it's able to make specific and precise changes from a C base to a T base. This method, as you might imagine, reduces the risk of any unintended and untargeted mutations, as well as allowing for precise editing. And again, mice edited with this technique showed longer and golden fur coat, with again some variation in texture, and as well as some mosaicism. Finally, the third method was embryonic stem cell editing. To overcome the challenge of mosaicism, where some cells get the edits while others don't, researchers decided to perform gene edits in MESCs, or mouse embryonic stem cells, in a dish. Then they injected these into an early stage embryo. This ensures that all cells in the organism carries the desired edits. And as we can see here, the edited mice show woolly traits. These edited mice show similar woolly traits to the previous methods. However, I think there's some unevenness in hair texture. If I had to rank them based on aesthetics, I think the first method is the most cute, followed by the second and then the third. And I think the researchers low-key agree because most of the press release images they use are mice from the first procedure. So what did we learn? This study demonstrates that CRISPR multiplexing is able to introduce up to 5 gene edits at the same time, while achieving an efficiency of over 90%. And indeed, these hair-related modifications gave clear phenotypic changes, demonstrating that they do indeed control hair length, hair texture, and pigmentation. However, the last edit, if you remember, involved fat metabolism. Well, researchers didn't see any changes in fat deposition or body weight. So in future studies, they want to challenge these mice with potentially a high-fat diet or placing them in a cold environment to see if they respond. And while this research marks a significant step forward in genetic engineering, there's a long way to go before bringing back the woolly mammoth. One consideration is that elephants and mice are separated by 24 million years of evolution. The changes we see in mice may not translate to elephants. The second consideration is an ethical one, as Asian elephants are considered endangered. And does de-extinction deprioritize any conservation efforts? And the final consideration is an ecological one, where mammoth-like elephants may have unintended consequences in their environment. We honestly have no clue what these gene edits in an Asian elephant to make them mammoth-like would do to the food chain, or their immunity, or their fertility. Well, for now, recreating a woolly mammoth seems like a distant goal, but the team at Colossal Research has placed their timeline for 2026. However, gestational periods of elephants are really out of anyone's control. In the meantime, though, we get these cute little mice, and who knows, they might become a thing in the future, but probably will be super expensive. I still want one though. Anyways, what are your thoughts on this research? Do you think de-extinction is a worthwhile pursuit? And which one of these methods do you think gave the cutest mice? Let me know what you think in the comments below. If you like this sort of content where I break down scientific research, please like and subscribe, and I'll see you next time. Ciao.